Hello students, in the last video we have completed the digestion of different types of foods that are carbohydrate, protein, fats and nucleic acid in the different regions of the alimentary canal, that is in the stomach, mouth, intestine, etc. So we have completed the digestion part. In this video we are going to see the absorption, then assimilation and then the next process, that is ejestion. So first of all, we need to know that whenever the food is being absorbed, how it will be absorbed, right? So we know that we have the villi, and this villi will have the evasination. That means it will have the elevations, then there will be a depression. So that depression will be known as the crypts of Leverkusen. Okay, so whenever the food is been suppose in the least size or it has been completely digested, then it will be present in the lumen. So how it will be absorbed? First of all, the cell membrane have to take the food particle into the inside the brush bordered epithelial layer of the intestine. Then after that it will enter into the lacteals. So these are the lacteals. So these lacteals are nothing but these are the lymphatic vessels, and then the along with that it will also have the blood vessel with it. Okay, so this way we are going to have the arteries and then we are going to have the veins too. Okay, so these are the veins, suppose. So veins and the arteries that will be connected by the capillaries. So eventually the food will be taken by the blood vessels, and the blood vessel will take the absorbed food material to the different tissues. So just see what happens if it comes to the different types of food particles, then how it will be absorbed. We know that the carbohydrate, the size, whenever it gets the minimum size or the unit, that is the monosaccharide. So that monosaccharide will include the three types of monosaccharides we have seen, that are the glucose, fructose and galactose. So glucose, fructose and galactose, these are the monosaccharides. Then for the protein, the unit will be the amino acid. For the proteins it will be the for the proteins it will be the amino acid, and for the fats it will be the fatty acid and glycerol. So how all these units will be absorbed by our body, let's just see all of those. First of all, just see the food molecule, that are the monosaccharides, that will include glucose, fructose, galactose, then the amino acid and the ions, that are the chloride ions, that will be diffused by the process of simple diffusion. So just see what is a simple diffusion. Suppose this is the membrane. Here we are going to have the high concentration of the molecules, and here we have the less concentration of molecules. Then always the movement of the molecules will be from the region of higher concentration to the region of lower concentration without the use of any energy. It is the natural law that the molecules, they will move from the region of higher concentration to the region of lower concentration. So the monosaccharides or the amino acids or even the chloride ions, that will move without using any type of energy. The region of higher concentration to the region of lower concentration, it will move. That is known as the simple diffusion. Now the another type of movement, that will be the facilitated diffusion, right? So what is the facilitated diffusion? See, first of all, glucose or the amino acids. See, glucose and amino acid, that can also be absorbed by the simple diffusion. Then same glucose and amino acid, that can be absorbed by the facilitated diffusion also. Now, what is the facilitated diffusion, right? So in the facilitated diffusion, suppose you have one membrane, right? So here from the region of higher concentration to the region of lower concentration, the molecules will not able to move. It will be by some transporter proteins, right? So just see, this is one transporter protein. Suppose this is the outside, right, and this is inside. Okay, so here we have one transporter protein. Okay, so this transporter protein is present like this. Okay, suppose this is one glucose molecule. So this glucose molecule will able to bind with the transporter protein this way. Then after that, what this transporter protein, there is one example of transporter protein, that is GLUT4. Okay, so for the transport of the glucose molecule, we are going to have GLUT4 on the plasma membrane of the cell, who is going to take the that suppose glucose molecule into it. Then what happened, these are the transporter proteins, they will flip, they will turn. So whenever it will turn, it will take the glucose molecule with it. Okay, it will take the glucose molecule and it will transport that unit or that glucose molecule from outside to inside. So this way the molecules will be accepted, that way the glucose molecule will be absorbed by the cells. Okay, so this way also the food molecule can be absorbed with the help of some transporter proteins. One example for the transport of glucose, I can give you the example, that is GLUT4. Remember, GLUT4. Okay, so this one will accept the molecules, bind with it from outside, it will flip, it will turn towards the inside and it will release the food molecule to the cell cytoplasm. So this is what this is called as the facilitated diffusion, right? Now after that, the third example you can see, it is for suppose water, right? So water, especially water that will be absorbed by our cell by a process that is called as osmosis. So what is osmosis? With just simple example I will tell you, otherwise the types of transport, the simple diffusion, facilitated diffusion, every type of process you will get in especially you will get it in botany. So every mechanism will get. Just for here, how the food molecule will be absorbed, you have to remember just the names. So just see, if it is suppose the water, you have one bucket, okay, you have water in it and you just drop one resin, okay, suppose it's a kismis. So if it is for a kismis, then what happen, you will just take it, okay, drop it in water. You just take that kismis into the water and just rest it for one suppose complete night. In the next day what will happen, this kismis will rise, isn't that? Why this is happening? Because from outside water have entered inside the cell. So this is just the process of osmosis, the movement of the water, that is called as osmosis. Through the osmosis process, the cell will accept the water molecule. And remember, water will be absorbed everywhere in the everywhere in the gastrointestinal tract, and maximum water will be absorbed in the large intestine. Next one, if it is the another type, once again movement, what is that? That is the glucose amino acid or the sodium ion that will be absorbed by another process, that is active transport. Okay, so what is this active transport? See here, for the active transport you require the utilization of energy. Okay, suppose here this is outside, this is outside, this is inside. Okay, in the outside you have less energy, less molecules, and inside we have more molecules. Then is it possible just by the simple diffusion the molecules here from outside to inside it will move? Never. Okay, so here what happens, we have to apply energy, and whenever we apply energy, the molecules which actually needed to be absorbed, even if it is less outside, then also we have to absorb it, because these are the digested food molecules and already we have higher concentration inside. In that case what happened, these molecules, even if it is less outside, it will apply energy, and with the apply of the energy it will be accepted by the cell, by inside the cell, this will be enter. Okay, this is not the natural law, but then also it will be by the utilization of energy. We will accept the nutrients, any type of digested food molecules to the inside the cell. So this is what, this is the active transport. So if you can remember that if it is carbohydrate rich, that is carbohydrate, glucose, or protein, that is amino acid, that can be absorbed by simple diffusion, by simple or facilitated diffusion, or active transport, depending upon the situation. So the next one, that is the fourth type of food molecule, how it will be absorbed, just see, fatty acid and glycerol. Okay, fatty acid and glycerol, what are they? They are the unit of the fat. So fatty acid and glycerol, how it will be absorbed, right? So just see, remember that fatty acid and glycerol, they are insoluble. Okay, if they are insoluble, they will not be able to absorb, right? So if it is not being absorbed, then what will happen? Just see, fatty acid and glycerol, they have this structure. This is the long chain fatty acid and this is the glycerol. Okay, glycerol. So this fatty acid is a long chain carbohydrate and this glycerol, that is the hydrophilic, and this fatty acid is hydrophobic. So this structure will not be able to absorb. So in that case, what it is going to do, it changes the configuration. Suppose we have a number of fatty acid and glycerol, it will make a comparatively large structure where the hydrophilic ends will be present towards the surface and hydrophobic ends that will be present towards the middle, okay, towards the center. So that's why from outside it seems like it is hydrophilic, right? So this large, that means these are the small drops which have the hydrophilic ends, that are the alcohol parts towards the surface, and centrally it is going to have the fatty acid or hydrophobic parts, that will be that structures that will be known as the miscellaneous. Normally the fatty acid and glycerol, they will be present like that. So whenever they need to suppose get absorbed, first of all it has to change the configuration, it has to change the structure. So that's why they will make a round structure where the alcohol will be present towards the surface and the center that will have the fatty acid. After that, just see what happens, that miscellaneous or the droplets, whatever it's present, that will be accepted by the cell's brush bordered epithelium. It is somewhere present here. These are the miscellaneous. Then those miscellaneous that will be that will be accepted by the lacteals. Suppose these are the lacteals, how it will accept that? The lacteals will not even accept the miscellaneous. Okay, so whenever they form they are already entered inside the brush bordered epithelium of the brush villi. Then after that, to accept the miscellaneous inside the lacteals, how it is going to be absorbed, right? So what happened? What we are going to have the fat droplets, right? So this fatty acid and glycerol, they form a somewhat hydrophilic parts towards the surface and hydrophobic parts, that are the fatty acid parts, towards the center. So after that, what it is going to do, this fat that will have a surrounding protein layer. Okay, so it will develop a protein layer. Okay, so whenever it will develop a protein layer, what will happen? That structure will be known as the chylomicrons, and whenever it forms the chylomicrons, then it will be able to easily absorb by the lacteals. And this chylomicrons, they are nothing but, remember, what is chylomicron? This can come. Okay, what is miscellaneous? That can come. Right, so these are very much important. You just please do watch the videos. They are going to be very much important. By just only reading or xeroxing the PDF format, you will not able to understand. You are not able to do all the questions that will come in the future. So that's why you have to watch the videos. Otherwise, why we're giving the videos, right? So the thing is, the miscellaneous, what are they? They are forming a hydrophilic parts towards the surface and hydrophobic parts towards the center. That are known as the miscellaneous. Miscellaneous will be accepted from the lumen of the intestine inside the brush bordered epithelium, and from the brush bordered epithelial cell to the lacteal, how the miscellaneous will be accepted. This miscellaneous will have a protein layer around the miscellaneous, and that structure will be known as chylomicrons, and now it has been permeable. So whenever that fat will have a protein layer around it, that will be known as the chylomicrons, and this chylomicrons will be absorbed by the lacteals. So this way the fats will be absorbed inside our body. So this way we have completed the process of absorption. After that, let us see move to the next process, that is assimilation. Okay, so what is assimilation? Assimilation is a process by which the cells take the absorbed food materials, that are the monosaccharides or amino acids or the iron or water, for its metabolism. Then this process will be known as the assimilation process. So assimilation is nothing but the process of utilization of absorbed food molecule by the cells. Okay, so that is called as the assimilation process. After the assimilation process, the next process will be ejestion. Okay, now what is ejestion process? Ejestion process means it is the removal of undigested food product. Okay, undigested food product will be removed outside by a process of ejestion. Now just see, where does the absorption take place, right? Absorption, maximum absorption takes place in the small intestine. Okay, remember, maximal absorption of the food will occur in the small intestine, right? Next, large intestine will absorb the water, maximum water and the electrolytes, etc. Stomach will absorb little amount of alcohol, and right from the mouth, if already we are taking, then absorption process will be started. Maximally absorption will occur in the small intestine. Now after that, the undigested food molecule, if it has been produced, if they have been suppose present in the small intestine, that will be pushed to the large intestine, and from the large intestine it will be stored in the rectum. Now in the rectum region, the undigested food molecule will be stayed. The muscle will give a stimulation to the brain. The medulla oblongata, they receives the stimulation. It produces a sensory impulse, and due to that we actually remove the waste particle by a number of action of muscles, the sphincters present in the anal structure, and from the anus we will just remove the undigested food molecule. That process is called as ejestion process. So this way we have completed the absorption of the food molecule. Assimilation and ejestion, just the process you have to know. So that's all about the process of digestion and absorption we have completed. In the next video we will see some hormonal regulation. So that's all about the video. I hope you understand. Thank you.